Dear students, in this unit, we will talk about how to address transgene escape. Now, before going to talk about this topic, you should all know that what is a transgene. Trans mean different, and gene mean a transfer of a gene in some variety of plant or animal which is not belong to that species, which is different, which is taken from another species. The example is, for example, if you introduce human insulin gene into the bacteria in order to quickly take a lot of insulin production, this is known as transgene. Now, the rapid progress of transgenic biotechnology has significantly promoted the development and production of GM crops. The same thing we are taking from the genetically modified crops. Crops are modified in such a way that the beneficial genes are either introduced into a species of plant to take benefits, or some harmful genes can be switched off, which may be not good for the humans or even for the plant. For the production point of view, those genes are silenced, or maybe a new genes are introduced which give them resistance against certain diseases. These all are being done at a very rapid pace in order to get higher yield, in order to cope with the increasing human population. Now, the cultivation of these GM crops, although they has yielded great benefits in terms of higher yields and more benefits, but at the same time, it has some serious biosafety issues, which can be a threat to the other natural species which exist in the same ecosystem. Now, when we talk about this gene transfer or transgene escape, that means that a certain species, one gene is introduced into that species, and when it is grown in a certain environment or in the outside in certain fields, there are chances, highly likely chances, that those transgene can be transferred or can be escaped from that species to the neighboring species or to the wild varieties. Now, the example of these are, for example, it has potential environmental consequences which are created by a possible transgene escape from these genetically modified crop to a non-GM crop counterparts. For example, it can transfer from a same crop to the wild crop, and it can also be transferred from one wild or weedy relative, mean from one crop to the wild crop via gene flow. And these are a very serious environmental concerns, and they have been extensively debated for biosafety issues point of view at international level. Now, gene flow is the movement of genes or genetic material from one plant population to the another plant population. You can see in this diagram, for example, transplastic pollen of Brassica napus. Normally, this pollen cannot be transferred to Brassica repa, which is the identical species. Now, when we talk about transplastic pollen or the species, actually in transplastic strains, the genetic modification is done in the plastids instead of the main genome which is present inside the nucleus. So the nuclear DNA is not modified. Instead, as you are aware that mitochondria and plastids, they normally have their own DNA. So when some changes are done in those DNAs of plastids or mitochondria, these are those organisms where these changes are done. These are known as transplastic species, plant species. Now, for example, there are certain species of Brassica. For example, this Brassica napus, it has this transplastomic gene. You can say it's a transgene. Normally, the pollens of Brassica napus, they cannot be transferred to Brassica repa. But in some other instances, for example, if Brassica repa pollen, if this Brassica napus, which is a transgene actually, it act as a female plant, and the pollen from Brassica repa, they are transferred to this Brassica napus. It becomes a hybrid or a plant like Brassica napus and Brassica repa, transplastomic hybrid. Now, this hybrid, if, for example, another Brassica repa, the pollens of those Brassica repa, if they are transferred to this hybrid of Brassica napus and Brassica repa, as you can see here, if the pollen of this are formed, which is formed as a result of the cross between Brassica repa, which is not a transgenic plant. Brassica napus is a transgenic plant actually. So when pollen of this are transferred to Brassica napus, the resultant species will be hybrid. Now, that hybrid species, again, if a wild Brassica repa pollen are transferred into this hybrid, then it has the ability to produce such pollens that those pollen can be transferred to Brassica repa. So you can see that there are certain ways that normally the transgene cannot be transferred from Brassica napus to Brassica repa directly. But when we talk about this pathway, after certain modifications and certain transgene transfers and the synthesis of certain hybrids, those hybrids can ultimately transfer that transgene to the wild Brassica repa, and that could be a very serious, according to the environmentalist or the experts which are related to the biosafety of these plants. Now, in another example, you can see here that, for example, we have some genetically modified plant which has a transgene, mean a gene which is taken from outside that species. Normally, if they are grown in a field, there are likely chances that their seeds can be mixed with some other species. For example, genetically modified maize, if it's grown in soybean crop, that can result in the transfer of that can result in the transgene escape. The same is that the oilseed rape in Japan and Switzerland, they have also resulted in the escape of transgene from the genetically modified plants to the other wild or the other weedy plants. Now, similarly, the variants of these GM plants, they can also have the ability to initiate the transgene escape. And similarly, the wild plant can also take the pollen seeds of these genetically modified plants. The examples are given in these, you can see here in these brackets, different examples of these transgene escape through different species, similar or the related species. So ultimately, this pollen-mediated gene flow from GM crop has resulted in the escape of transgene from those genetically modified plants to the wild or the weedy species. Now, this is a very serious issue, how this transgene escape can be addressed. Now, certain act can be done, or certain measures can be taken. For example, a site-directed mutagenesis can also be done. Site-directed mutagenesis is actually a type of mutation in which a typical allele or typical gene of one plant is targeted and mutated in such a way that this mutation is very unlikely to escape or to transfer from this species to the neighboring or to the other species. And there are some other methods like cisgenic strategies. In the cisgenic strategies, the expression of certain genes is tightly regulated in such a way that if it cannot be transferred to the other crop populations. And by doing these things, this transgene escape can be avoided. Now, in certain cases where transgenic trait is needed, there should be efficient containment using standard approaches. They should be applied so that to ensure that there is no transgene escape. For example, in certain studies, they have made sure that if a transgene or a GM crop is grown in a field and more than 20 feet or 20 meter away, the other crops are grown, there are very less likely chances of transgene escape. So these standard measures can be utilized in order to ensure to avoid maximum transgene escape. Now, these cautious approaches using these novel type of GM crops, it will not only enhance the sustainable potential of these GM crops, but it will also increase the public interest in the green gene technology. When people will be aware that no gene can be transferred from that GMO to the other species, people feel more comfortable that it is very safe to consume, and more people will start trusting this GM crops and this gene technology, green gene technology. Now, when we talk about the flow of gene, for example, transgene has escaped from this genetically modified crop to a wild one. There can be multiple ways of this gene flow or the gene transfer. It could be vertical gene transfer or vertical gene flow, or it could be horizontal gene flow, which is also known as horizontal gene transfer. Now, vertical gene transfer, as you are aware that from parents to the offspring, when a gene is transferred, it is known as vertical gene flow or vertical gene transfer. While horizontal gene transfer, it normally occurs between unrelated species. For example, a gene from a microorganism is transferred to a fungal species or a plant species in their root nodules. This is known as horizontal gene flow. Now, these when we talk about the avenues of gene flow, there are mainly three avenues. It could be transferred through pollen or seed or even vegetative propagations. So this can be minimized by using biosafety approaches, and it will increase the trust of people towards this genetically modified crops.